And by the way, it's multiple choice, so bring the pencil. Oops, wait a minute. Okay, any questions about that? Okay, so let's go ahead and get started on the quiz. And I'm going to say, what it says. Go ahead and answer. Double click or make sure that you voted. Sometimes it doesn't get recorded, so you want to make sure you did. Go ahead and double click it. Got everybody. All right. See what we got. Did I stop it? Well. Oh. I actually turned off my thing. Okay, that would help. I had it on. Okay. All right. So we've got a little bit of everything. What you want to remember is gustatory receptors. They are those little taste receptor cells in the taste buds. They stimulate first order sensory neurons by releasing neurotransmitters, which means they're receptor cells. They're not the first order sensory neurons themselves. They are receptor cells, which means that they generate, when they get stimulated, they generate receptor potentials. Receptor potentials are those depolarizing graded potentials that are generated when you stimulate a cell with a stimulus, but it's got to be, the receptor cell. So if it were a first order sensory neuron being stimulated, it would be a generator potential, which generates action potentials in the neuron. But these are receptor potentials, which would be D, and that is a depolarizing graded potential that stimulates the release of neurotransmitter, which in turn stimulates the first order sensory neuron. So it's a deal there. Okay, so here's the next one. Why sometimes it works, sometimes it doesn't. Okay. Go ahead and make a choice. Double click. Especially if you see a check mark when you voted. There should be a check mark if you vote and and it receives it. Okay, so raising the upper eyelid is correct. If you know levator means to elevate, and then palpebrate is the eyelid, it's raising the upper eyelid. Focusing the lens is done by the ciliary muscle. Pulling the eyeball superiorly is the superior rectus, which is one of the external oblique muscles. Closing the eyes would be the orbicularis oculi, and raising the eyebrows is the frontalis muscle. So, okay. Any questions about that one? Okay. Next one. vote. Double click it, okay. All right, sclera is correct. The whites of your eye is the sclera, and it's collagen that makes it so white. And here's the last one. One more vote. Double click it, okay. And let me see. Cone photopigments bleach and regenerate in bright light. Yes, this is correct. B was correct. We'll look at the choice there for a minute. Rods, remember, are only active under low light conditions, which would be at night or really dark conditions. If it's bright light, their photopigments bleach. It's also called photo bleaching, which means bleaching because of that light exposure. They photo bleach in bright light, but they don't regenerate until it gets dark again, which is why you have to let your eyes recover or adapt when you go from light to dark. It takes a while for those rods to regenerate their photopigments. Cones can bleach and regenerate during bright light conditions, so they're active under bright light, and they can see color. They're able to regenerate, and they're able to do so very quickly, so they can remain active even under bright light conditions. But during low light conditions, there's not enough light to activate them, so they don't respond to low light. That's why when you're in dark, everything's black and white or gray. Any questions about that? And I'm going to find, I thought I had a website up on my previous Blackboard shell, but it's not there. But I'm going to try to find a website about color vision and about color blindness specifically, and I'll post that for you when I find it, because there's some good websites about that. Okay. Now we're going to finish up the special senses, and let's see. We're on to sound waves and hearing. So let me get over here.
It's like OJ.
Okay, this is just showing you a picture of the external auditory meatus in the temporal bone. So let me move on, but that's the location of the ear canal, right behind your temporomandibular joint, or jawbone, or jaw joint. Okay, so we were talking about this already, so let me move on and finish this up. I want to try to quickly get through the rest of this so that we can get to the endocrine system today. So I don't get even more behind than I already am. So we're going to talk about how sound waves work. And the sound waves travel, first of all, the auricle, which is your outer ear, helps in funneling sounds to your ear. And that's why some animals, they have outer auricles or pinna that can be moved so that they can capture those sound waves better. Ours don't move around very much, but it does help to kind of funnel the sound waves into the external auditory meatus, that opening in the side of your head there. And then the sound waves are going to vibrate the tympanic membrane, which in turn is going to vibrate the little ossicles, the auditory ossicles, which are teeny tiny little bones. They're very, very small. Teeny bones in your middle ear. So this is the external ear here, the external auditory meatus and the external ear here. And then behind the tympanic membrane, which is your eardrum, is the middle ear. And that's where you have the auditory ossicles here in yellow that transmit sound waves to the organ of hearing. The blue here, this part, is where you have the organ of hearing, the cochlea, as well as the semicircular canals and other part of the vestibular apparatus here. But right now we're focusing on the cochlea, which is where your organs of hearing are located. And again, the middle ear is connected to the eustachian tube or auditory tube that connects to your throat. So that's the middle ear. And let's look at technology today for me is not working very well. I've had all sorts of computer troubles today. But anyway, this is a magnified image of your tympanic membrane, your eardrum, which spans that part of the ear canal at the end of the external auditory meatus, which is your ear canal. And so sound waves vibrate that, which in turn vibrates the malleus. The malleus is supposed to be shaped like a mallet or hammer. That's what it's named for. So that's what malleus refers to. That's the Latin term for hammer or mallet. And then the malleus, in turn, it vibrates and makes the incus vibrate. Incus is Latin for anvil. And I guess somebody thought it looked like a blacksmith's anvil. I don't know about that, but that's what that's named for, the incus. And then the incus makes the stapes vibrate. Now the stapes means the stirrup. It does look like a stirrup on a saddle. So that's the only one that really looks like what it's supposed to look like. But maybe that sort of looks like a malleus or a hammer. But anyway, this is the stapes, and it covers a part of the cochlea called the round window, and that's, the oval window. There's also a round window, but we're not going to worry about that. But the oval window is where vibration is transmitted from the stapes to the cochlea. And we're going to look at that in just a moment. I do want to point out that there are some muscles. Why this isn't wanting to work. Okay. There is a muscle attached to the stapes. And where is, okay, so here's tympanic membrane, malleus, incus, and stapes. And if you look at a close-up view of the stapes, there's a muscle attached to it called the stapedius muscle, named for the stapes. And when you hear loud sounds, really loud sounds that make the bones vibrate too much, that muscle contracts a little bit, tightens the stapes a little bit to kind of dampen some of that vibration to prevent nerve damage. So there is some mechanism of trying to prevent overstimulation of your spiral organs that are involved in sound, and we're going to look at that in just a minute. So there's a description of that stapedius muscle. Maybe I can get this. This is going nuts. Okay, let me try something real quick. I'm just going to reopen my iClicker. Use that. It's not. That other thing's not responding. Okay. So we're not having another quiz. I'm just going to use this to advance these slides. Okay, that should come up pretty soon. I might get rid of that. Okay, so this picture shows, or the next picture I should say, shows in more enlarged view of the stapedius and how it's attached to the stapes. Anyway, from that point, the sound is being transmitted to the cochlea, which is part of the membranous labyrinth. Now, the membranous labyrinth is found, it's a membrane-bound system of channels, and it's enclosed within the bony labyrinth, and inside your inner ear, this is the inner ear we're talking about, you have a bony space that fits the shape of the membranous labyrinth. The membranous labyrinth being the vestibular apparatus and the cochlea. And so there's some fluid in there. It's called perilymph. Don't worry about that fluid in particular, but there's fluid between the bone and the membranous labyrinth. The membranous labyrinth itself has a membrane around it that encloses these channels that contain endolymph. You do need to know that name, endolymph, which is inside the membranous labyrinth. And it's the endolymph fluid that helps in the functioning of transmitting sound waves and the functioning of your equilibrium receptors. So there's 3 parts of the membranous labyrinth. Again, these fluid-filled spaces within a membrane. The cochlea, where your organs of hearing are located, semicircular canals, which are involved in equilibrium, and the utricle and saccule in what's called the vestibule, and these are also involved in equilibrium. We'll talk about those in a little bit. But anyway, the sound waves, or the waves from sound, the vibrations, I should say, are transmitted by these bones to the oval window, which is underneath the stapes. It would fit that shape there. And now it's being transmitted to the cochlea. You can kind of see it dimly outlined here because they're kind of highlighting just the middle ear there. So here is the membranous labyrinth. So what they're showing here is there's bone that fits around the membranous labyrinth in pink. The bone contains fluid between here and here, so there's some fluid there. And then the membranous labyrinth is the pink structure and yellow structure, and it's all filled with endolymph fluid. And that fluid moves, and that's what creates some of the functioning of this structure. So here's the stapes, and it fits right about here at the oval window. It is now transmitting vibrations into the endolymph fluid, and this is the cochlea. Now, when we talk later about what's going on in the cochlea, the apex, this would be the base of the cochlea, and this would be the apex of the cochlea right here, the very end. If you were to unroll it, that would be at the very end of the line. So these vibrations can travel through these canals or these channels, to the apex portion. And I just point that out because there's a picture that straightens it out. You kind of have to know what you're looking at. So we're going to worry about this other stuff later, but let's look at what's going on. So this is showing that this is showing the direction of those vibrations moving through the fluid. What's actually happening is once you get into the cochlea, the vibrations from the stapes cause pressure waves to form in these canals. Pressure waves, which are sort of like vibrations, but within the fluid itself. So the fluid is vibrating. And so what happens is, and you can see this when they cut it open, you can see these canals. Now there are different kinds of canals. We're not going to worry about the names of the canals, but it's those pressure waves in the endolymph in these canals that vibrate the spiral organ and stimulate what are called sensory hair cells. That these are hair cells that have little hair bundles on them, and those are what detect vibration created by sound waves. So this one, this is just showing you what I just showed you. This picture, they've cut it open in kind of a cross-sectional kind of cut, so you can see these different fluid-filled spaces like that. And in these fluid-filled spaces, let me see here, you've got, you can see that there's little partitions in between. And it's in some of these partitions that you have what are called the spiral organs, which are the organs of hearing. Spiral organs consist of hair cells in these regions that are stimulated by the pressure waves in the endolymph fluid. So this is just showing you the direction that the vibrations are going up to the cochlea. And let's look at what happens from there. So this has been kind of partially unfurled. In other words, it's not as tightly coiled in this diagram. But this is showing that with the vibrations, you can see looks like a little movement there. The vibrations are transmitted into the endolymph fluid. And, actually the endolymph fluid is in here. And this vibration in these fluids, or in the fluid here, is going to stimulate, let me find the base, okay, there's the fluid. the basilar membrane right here is being stimulated or vibrated, and that in turn vibrates the spiral organ right here. And then there's another membrane, we're going to look at the vestibular membrane right here. So the spiral organ lies between two membranes, the basilar and the tectorial membrane. So we're going to look at what is going on right here at this level. And it's just, forgot how to click through that. So anyway, that's just showing the move. You know, you can go to your book and each step is discussed. I don't go into as much detail as your book goes into though. Okay, so let's look at what happens in the spiral organs. So the spiral organs are these organs of hearing between these fluid-filled spaces. So, I'm sorry, I was calling it endolymph, it's actually perilymph in these spaces, but it doesn't matter, it's a fluid-filled space. So, here is something I'm going to magnify for you. This is part of the vestibulocochlear nerve. This is actually the cochlear branch, which carries sound information to the vestibulocochlear nerve and to the brain. So information is being transmitted this way. What we're going to look at is a magnified image of this structure in the next picture. And here you can see the basal membrane, or basilar membrane, which is at the bottom of this structure. And then you have the tectorial membrane, which is kind of a gelatinous membrane. So the basilar membrane, if you're looking at it up here, is, well, it's kind of hard for me to point to that. Right there is the basilar membrane that they're showing here. And then at the top, you have this kind of gelatinous the tectorial membrane. And sandwiched in between are some other cells we're not going to worry about, but there's hair cells. And these hair cells have tiny little hair bundles, which are what are called stereocilia. And stereocilia are like cilia, but they're not the kind of cilia that wave like you have in your windpipe. They are more, you know, just they do move, but they're moving in response to vibration, so they're vibrated. So when you have pressure waves in the fluid inside your those channels, that vibration that goes on, that pressure wave or that vibration in turn vibrates the the basilar membrane, which in turn vibrates these guys and makes them move, vibrate back and forth. So they're little hair bundles. I don't know if you can see them very well, but on your own computer you could probably see them. It's a little yellow thing sticking up there. Those little hair bundles start vibrating against the tectorial membrane. So it's like these are your hair bundles, and this is the hair cell and the hair bundles, and this is the tectorial membrane. They're vibrating against that. And that's what generates receptor potentials in those cells. Those cells in turn, these are receptor cells, so they're going to release neurotransmitter that is going to stimulate first order sensory neurons to transmit impulses to the brain through the cochlear branch of the vestibulocochlear nerve. Don't worry about inner or outer cells. I'm not going to get into that much detail, but those little hair cells are being vibrated against the tectorial membrane. They're going to then depolarize. Those cells are going to depolarize and in turn stimulate first order sensory neurons, which would be found in the cochlear branch of the vestibulocochlear nerve. So let's go on to the next. And so here's a really big, here's that really big picture of it. I don't know if this is any better, just maybe from a different source. But anyway, here you can see, see them pretty well. You can sort of see those little stereocilia. And then here's one of those, and here's some, again, sensory neurons transmitting information through the cochlear branch. There's actually two branches of the vestibulocochlear nerve, which is nerve number 8, cranial nerve 8. There's the cochlear branch, which transmits information from the cochlea, and then there's the vestibular branch coming from the equilibrium receptors. This picture is kind of a schematic showing you what I just showed you. So here they've straightened out the cochlea. So there's the apex of it right there. And again, I'll go into some of the details that are shown here, but here is the basilar membrane. The organ of Corti is the same thing as the spiral organ. It's those hair cells, basically. And then those hair cells are vibrating against the tectorial membrane. So this is sort of sandwiched in between these two canals here. And that in turn is going to generate impulses that are transmitted through the cochlear branch of the vestibulocochlear nerve. Okay, so I'm not going to worry about these receptor potentials. It's basically, well, I guess we could talk about it very briefly because this is something we've talked about before. Any kind of mechanically stimulated sensory cell is, when it's mechanically stimulated, and that would be like touch and pressure in your skin, but also sound waves in your ear, that opens by that vibration of those little stereocilia, that opens up mechanically gated potassium ion channels, and they're called transduction channels because you're transducing a stimulus to a receptor potential, and so you're getting those depolarizing receptor potentials. So we've just talked about all that. But that's how you get those receptor potentials. It's a potassium ion channel, potassium flows in and depolarizes that cell. It in turn releases neurotransmitter, which stimulates action potentials in that first order sensory neuron of the cochlear branch of the vestibulocochlear nerve. And this shows from that, there's a little opening in the cranium, and through that opening you've got the nerve carrying information to the thalamus and to the primary auditory area of the brain. Some of that information crosses over. So you do have some of that information going to the same side of the brain, some of it crossing over to the other side of the brain, and that helps you put together sound from coming in from both sides of your head. Your brain is putting together those sounds. The primary auditory area, there's a more enlarged view of that. The primary auditory area, well, we'll see that in a second. I just want to put this picture in so you can see where the internal auditory meatus is. This is where the vestibulocochlear nerve is coming in. The organs of hearing and equilibrium are in this bone here. So that's where those nerves are passing into the cranium. Here's the location of the primary auditory area, which is also surrounded by the auditory association area that allows you to recognize what you hear and store sound as memory. You might notice that the auditory area is close to the speech area. So you hear something and then you quickly respond to what you hear. The information transmits back and forth. It's very close together, so you can have very fast transmission of what you hear in a conversation and your response back in terms of the Broca's speech area, allowing you to talk back. Now this complicated looking picture, don't freak out about it, it's just showing you the cochlea unfurled. This is the apex of it at the end. And what it's showing is that different regions of the cochlea are sort of tuned or adapted to hearing certain frequencies. In other words, remember that sound waves, you have different frequencies of sound. High-pitched sounds have very short wavelengths, high frequency, and then lower sounds, lower-pitched sounds, have very long waves, sound waves, and lower frequency. So notice that the higher frequency sounds are picked up at the beginning here, at the early part of the cochlea. So these cells are more going to be responding to high-pitched sounds. And then the lowest pitched sounds are detected way down the line at the end of the cochlea. So at the end of that, end of the curl. And then middle pitched sounds are in the middle. This is why people lose their high-pitched sound ability as they age, because as you age, you're exposed to all these sounds, especially loud sounds. Loud sounds cause too much vibration. Even with that stapedius muscle, you get too much vibration going into your ear, and so that strong vibration, vibrates those little hair cells too much and tears up those little stereocilia, and so as a result, you lose high-pitched sound ability to detect high-pitched sound first, usually, as hearing loss progresses. That's why you want to protect your ears from loud noises so you don't lose that high-pitched sound ability. But people do tend not to hear the higher pitched sounds as they get older because of that. And there's another picture here. So this is yellow is the high pitched sounds. The red is medium pitched sounds. The blue is lower pitched sounds. So that's the end of the line there. That's the apex. And this also shows that the primary auditory area, different parts of it process those different pitches. So, here's high pitch, medium pitch, low pitch. They're adapting, so they're picking up and processing that different pitch information. But it gets all put together by the auditory association area. That experience of sound gets put together by the auditory association area. So this is where you'd have high pitch sounds. This is where you'd have the lower pitch sound ability. Or that's where you would detect it. Okay, so moving on to the vestibular apparatus. Any questions about sound? Detection of sound. Okay, so the rest of this here in pink, now this is the part that actually contains endolymph. I forgot that this is perilymph and endolymph, and I'm not going to worry about those fluid names. Just know they're fluid-filled spaces because there's plenty of other stuff to worry about. But in these regions, these are involved in equilibrium, which we think of as being balance, but it's the ability, equilibrium is the ability to detect body position and movement and help maintain balance and all of that. So you have different regions of the vestibular apparatus that monitor equilibrium. There's two kinds of equilibrium, static and dynamic. Static is the detection and maintenance of your head position when your body is stationary. What static equilibrium does is it notices head movements, just your head moving, and not like walking around kind of movements, but when you bend over or your head moves down or back, that kind of movement is static. Dynamic is more in response to acceleration and deceleration of the head. So if you're walking or moving in an elevator or making more of a movement, that's dynamic. And so altogether, these structures, the utricle, sacral, and semicircular canals, are involved in maintaining static and dynamic equilibrium. And we're going to look at the utricle and sacral first. The utricle and sacral, here's the utricle, it's a little bulge right here. The sacral is over here, and together these form what's called the vestibule. I'm not so concerned about the vestibule, but utricle and sacral you should know. And that's just above the oval window. Let's look at those first. They are involved in detecting information about head position as well as the head moving through space, linear acceleration, moving forward, backward, or like when you bend over or move back. And we'll worry about the semicircular canals in a minute. So let's look at what's in the sacral and utricle. You have, there's actually a better picture of this somewhere. Here we go. Okay, well, you have the sacral and utricle have these structures in them, and they're at right angles to each other. So one runs this way, one runs this way, so that they can detect head movements. And these structures contain hair cells in the wall of these structures, little hair cells that have stereocilia in them, which are, again, these structures that can be bent as fluid moves past them. And they are covered in a gelatinous membrane called the otolithic membrane. And I don't know if I have a bigger picture of that. Yeah, there we go. So here's one of these structures. This is called, these structures are called the macula. And here's the otolithic membrane, and here's those little hair cells. So these are receptor cells. And they're kind of similar in appearance to the hair cells in your cochlea. Their stereocilia are a little bit different. That's not a big deal. But what they're covered in is a gelatinous material that contains calcium carbonate crystals. So those calcium carbonate crystals can pull on the hair cells because it kind of creates some force of gravity. So let me go back to a couple of these pictures. For example, in this guy's utricle or saccule, you've got these little hair cells covered in the autolytic membrane. There's the little otoliths, which are the calcium carbonate crystals. And right now, everything's at rest. But when he tilts his head down and looks down, that pulls on those stereocilia and stimulates those hair cells. And again, these are receptor cells, so they generate receptor potentials. And then they, in turn, stimulate sensory neurons to send impulses to the brain. So because you've got the force of gravity pulling on those calcium carbonate otoliths, that pulls on that otolithic membrane, which in turn pulls on the hair cells. So here's a picture of the hair bundle. You've got what's called a giant kinocilium, and then these smaller stereocilia, and the direction that they're pulled, depending on if you're like putting your head down or putting your head back, determines what kind of action potentials are generated. And I've got a picture of that somewhere. But there's the actual photograph of what those stereocilia look like. And again, here's the enlarged picture. But here's another picture showing you kind of the same thing. Here, when she's just standing upright, not moving, the otoliths are at rest. But when she puts her head down, the otolith sags down and bends those stereocilia. And this would also happen in the opposite direction if she tilted her head back. Here's a guy bending over, standing upright, nothing happens. There's the otoliths, there's the hair cells projecting up. He's bending down, those otoliths pull down and bend those stereocilia, which is what stimulates the opening of potassium ion channels and stimulates receptor potentials in the hair cells. The direction that the hair cells get pulled determines the type of action potentials that are generated in those first order sensory neurons that they stimulate. So if you pull the hair cells back, which I think is when you pull your head back, you're going to get, let's see, actually this one is, I think, pulling the head forward. You're going to get more action potentials. If you tilt your head back, you get fewer action potentials. So the direction that the stereocilia are pulled is going to either cause more stimulation or less stimulation, and your brain, and then this is being transmitted to your brain through sensory neurons, and your brain is going to interpret that as meaning you're moving your head in one direction versus another direction. So there we go. So I was just showing you the same thing here. So the direction that it's pulling, and in this case, pulling your head forward, I believe, is the one that causes more action potentials to be generated. If you pulled your head back, you'd have fewer action potentials. Now, if your head is perfectly still, there'd be no action potentials. So your brain is interpreting, fewer action potentials, tilting back, more action potentials, tilting forward. Your head perfectly still, no action potentials. That means your head's still. So your brain is interpreting that information. Okay, so moving on to the semicircular canals. They don't have otoliths, but they do have this endolymph fluid in them, just like the utricle and saccule does. So here's the utricle and saccule, where you have those otolithic membranes. Semicircular canals are similar. You have this structure called the crista, which consists of a mound right here, that mound of cells, and in that little hill within the semicircular canals. And here's the location of these structures. There's fluid here moving around, but this is where the sensory neurons are in the blue area, right at the base. It's called the ampulla, or base, of the semicircular canals. And here's some hair bundles similar to what you see in the utricle and saccule. And they are not covered by an otolithic membrane, but they are covered by a gelatinous cupola. It's like a big hat that sits on top of them. And as fluid moves past, it pulls on the cupola one way or the other, bending the hair cells opening up mechanically gated potassium ion channels, and then, you know, depolarizing those receptor cells, which in turn are going to release neurotransmitter and stimulate the first order sensory neurons to send signals to the brain. So they show a couple pictures of this. Here's one where she's turning her head to the side, and that's causing the fluid in those semicircular canals to move in that direction, and is pulling on the cupula, which pulls on the hair cells, which then, you know, generates action potentials. So when you're doing, you know, rotational type movement, when you're spinning around, turning your head, that kind of thing, you're activating those receptor cells. So let's see if there's talks about it anywhere here. So rotational movement as opposed to linear movement is what semicircular canals are involved in. I forgot to mention that, notice how they are projecting in different planes. One projects up, one projects back this way, one, you know, and come back and out, and one is more horizontal. So depending on the rotational movement you're making, it's going to cause different kinds of movement of fluid in the semicircular canals. And the ampulla here, here, and here is where those receptor cells are, where those cristae and cupulas and hair bundles are, or hair cells are. I may have another picture that shows this, but again, as you bend those hair cells, you generate receptor potentials in hair cells, which then stimulate sensory neurons to send signals. This time through the vestibular branch of the vestibulocochlear nerve. That's that same picture. Here's another one, kind of showing you the same thing. Moving the endolymph in the semicircular canals bends the cupula. And that information gets transmitted, again, the vestibular branch merges with the cochlear branch, and the vestibular branch sends signals to the other side of the brain through the thalamus, so the signals cross over to the opposite side of the brain where your vestibular processing happens in the primary vestibular area, which is near the primary auditory area. I don't think there's a picture of it on the brain picture. We're not going to worry about that, but it's fairly close to, it's there on the side of the head, close to the primary auditory area. So that information does cross over as opposed to some of the other special senses. Okay, this gets into a bit more, it's a little bit complicated looking, but this is just showing that equilibrium isn't just coming from your equilibrium receptors in your inner ear, it's also coming from your eyes and from your proprioceptors, and there's a lot of different processing of this information, but one of the major processors is the cerebellum, which is processing visual information, equilibrium information coming from the vestibular apparatus, and this would be here proprioception. All of that's being processed to help you maintain your balance. Oops, seems like I had another picture, but maybe that wasn't, I guess not. But anyway, if you go back to the cerebellum picture, which I thought I had in here, that's weird, maybe I accidentally deleted it. But if you go back, let me see if I can find it real quick. If you look at the cerebellum picture, back at the end of vision, I think. Where is it? Somewhere in here. Too many pictures. I was trying to take out some pictures and move them around to make this more easy to follow, and I was having computer problems this morning, which has completely thrown me off because I couldn't save anything and I couldn't put it on Blackboard either. So I'll get this, I'll maybe put some additional information on Blackboard for you. But, oh, that's it. There it is. So cerebellum, where it talks about the vestibular apparatus, that's the semicircular canals and the saccule and utricle. Okay, and I think that's it for senses. Any questions about vestibular apparatus? Okay, or sounds? So we'll move on to the next thing, and that is the endocrine system. Now, everything up to this point is on exam 1. What we cover today and Thursday will be on exam 2, but we have to get a head start because we are behind. There's a lot of information, but I'm trying to shorten up or make the endocrine system flow a bit better. But again, because I was having computer problems, I wasn't able to save my newest version of the notes, which are easier to follow and more succinct, but I'll do the best I can with these notes. So keep in mind, I haven't posted my notes either because I wasn't able to, my PowerPoint just kept giving me the spinny rainbow this morning, so I couldn't post that on Blackboard, but I will get that posted for you, hopefully today. Hopefully my computer will start behaving. So anyway, we've already talked, we've talked about the nervous system, and that's, last semester and this semester. Now we're going to look at the endocrine system. The endocrine system, instead of using neurotransmitters and action potentials and all that to transmit information, it uses hormones, which are chemical messengers that are usually released into the bloodstream, sometimes not. We'll see some exceptions to that. But released out of the cell, these hormones bind to target cells, which are usually far away. Neurons stimulate other cells directly in synapses, but endocrine cells release their products, their hormones, usually into the bloodstream, and so it takes for that blood to carry the hormone to the target cell, a few seconds, but it's going to be a slower response compared to the nervous system. Nervous system, you get a quick response, automatic response, but also a short-lived response. When I stimulate my muscle to contract, I don't keep contracting for an hour. I just, I do it and I'm done. Endocrine system can have very long-lasting effects. Those hormones their effects can last longer, but it takes a little bit longer to get the response. Sometimes it might take minutes or hours or even days to see a response in the endocrine system. And here's a comparison of the two. I'm not going to spend time on it, but you can kind of, look in your own book or on this table to see some of the comparisons between nervous system and endocrine system. Now, again, one other thing about it is the target cells in the nervous system are limited to muscle cells like smooth cardiac and skeletal muscle and glandular cells and other neurons. Endocrine system has the body, so it doesn't stimulate glandular secretions. It doesn't just stimulate sensory information, or doesn't just generate sensory information. It doesn't just stimulate muscle contraction. It has a lot of effects, as you'll see. Now there are, hopefully you remember, two types of glands in the body, exocrine and endocrine. We're not talking about exocrine glands here. Exocrine glands have ducts and empty into body cavities or the body surface and secrete a wide variety of products like sweat, oil, bile, milk, and things like that, and saliva, all that stuff. These are not endocrine glands. These are not hormones. So this is not endocrine. Endocrine glands are ductless. They do not have ducts. They're called ductless glands, meaning no ducts. They secrete their product into the interstitial fluid around them, and that hormone usually moves into the bloodstream. No ducts, okay? So here is an example of how endocrine glands work in a very simplistic way. Here is an endocrine cell in a gland, releasing by exocytosis in this example, releasing its hormone to the interstitial fluid around it, which is the tissue fluid. But that hormone is going to diffuse into capillaries. Capillaries are the tiniest blood vessels. It's at the capillary level where exchanges happen. Arteries and veins don't pick up hormones, capillaries do. And these tiny capillaries pick up the hormone and carry the hormone away to a far off, distant target cell. It might be on the other side of the body. The hormone exits the blood, diffuses out of the blood, and binds to a hormone receptor on the target cells, and that generates a response, a physiological response. We're going to talk about a little bit about that in more detail in a minute. In order to be a target cell, you have to have a receptor for that hormone. So, for instance, insulin, if this were a pancreatic cell, and this were insulin, insulin hormone, which is what lowers your blood sugar by stimulating cells to take in more glucose into the bloodstream, most cells in the body have insulin receptors on them. So in the case of insulin, just about every cell, probably every cell in your body has insulin receptors. And so if insulin binds to the receptor, the cell takes in more glucose, and other processes happen as well. But if this were, say, a different hormone, it could be, let's say, testosterone being released by cells in the testes. Testosterone is not going to affect every cell in the body. It's only going to affect cells that have testosterone receptors on them. And that's going to then generate a physiological response in those specific cells. So cells have to have receptors to respond to the hormone. Without a receptor, they don't respond. Now, there are some exceptions to the whole thing about going into the bloodstream. There are what are called local hormones. For example, there are hormone producing cells called paracrine cells. These are endocrine cells that release their hormone to a nearby target cell. It can be right next door. So it doesn't go through the blood. It just gets released and binds to a nearby cell in the same tissue, and so it's considered to be a local hormone. There's even what are called autocrine. Auto means self-stimulating. What that means is it releases a hormone that binds to receptors on its own cell membrane, stimulating a response. And you might think, well, that's weird, but it happens. There are some autocrine cells in the body. So it does seem like an odd kind of way to stimulate a response, but autocrine cells will stimulate responses in themselves to get, a particular physiological response. But most of the hormones we're going to talk about are the regular type of hormones. We'll, touch on a few local hormones, but mostly we're going to be looking at the regular type of endocrine glands. And those include the pineal gland, the pituitary gland, both of those in the brain, the thyroid and parathyroid, which are in your neck, around the trachea or windpipe, the ovaries and testes, the adrenal glands, the pancreas. Some of these endocrine glands also have exocrine function, like the ovaries not only produce progesterone and estrogen, but they also produce the egg. They release an egg. Testes not only produce testosterone, but they release sperm into tubules, so they're both exocrine and endocrine. And we're going to really focus on those when we get to the reproductive system. I'm going to kind of skip the ovaries and testes because that's more reproductive system stuff we're going to talk about there. Adrenal glands, we're going to get to those. Those are on top of the kidneys. The pancreas, though, the pancreas is also both exocrine and endocrine. It produces hormones like insulin and glucagon, but it also has a separate group of cells that release a substance called pancreatic juice that goes into the small intestine, and pancreatic juice is used for digestion. It contains digestive enzymes. So some organs or some glands can be both exocrine and endocrine. They have separate endocrine cells and separate exocrine cells, and that's the case with the pancreas. There's also some organs in your body that are not really endocrine glands, but they do have scattered cells in them that produce hormones. Many of your digestive organs, like your stomach and small intestine, have hormone-producing cells in them. We would not consider the stomach or the small intestine a gland. They're obviously not, but they do have some endocrine cells in them that produce hormones. Your skin, your heart, your thymus, those are the kidneys. These are organs that do have some endocrine cells in them, kind of isolated cells that produce hormones. We're not going to talk about all of them, we might touch on a couple of them. Also the hypothalamus, almost forgot. Hypothalamus, we've talked about that in terms of like autonomic system function and things like that. The hypothalamus in the brain is associated with the pituitary gland, and even though it's a nervous system structure, it's part of the brain, it also is a hormone-producing structure, and that's what we're going to be getting to pretty soon. So here's a list of some of the structures that are not glands. Even fat cells produce hormones. So these are cells, they're organs that produce hormones as well. And let me see. Okay, so we've talked about receptors. I talked about it in terms of that picture that you have to have a receptor on the target cell. These receptors are continuously being produced and also broken down as needed. So target cells can be stimulated to produce more receptors, or they can be stimulated to produce fewer receptors. But we're talking not just a couple of receptors, thousands of receptors on a single target cell. Now, this is one of the things that I was trying to fix because there's a lot of stuff on that page. So one of the things I was trying to do is simplify these slides. But like I said, once I get that fixed on my computer, I'll post it. But anyway, there's two ways that you can regulate the production or breakdown of hormone receptors. Okay, like I said, going back to that previous picture, receptors are being synthesized and broken down as needed. That's what upregulation and downregulation refers to. Upregulation means that a hormone, you start producing a hormone, you're not producing them all the time. You only produce hormones as needed. So sometimes you're producing a particular hormone, sometimes you're not. But if you're producing a hormone, certain hormones, they will stimulate target cells to start making more receptors and putting them on their cell surface. Cells can put more receptors on the cell membrane, on the plasma membrane, and that's called upregulation, and this makes them more sensitive to that hormone. So when a cell is upregulated, it's going to start expressing more receptors. Now, when I say receptors, some receptors are internal inside the cell, some are on the cell surface. We'll talk about that later. But there are some hormones that stimulate upregulation, getting more receptors, which makes them more responsive to the hormone. And then other times when that hormone's not present, then you'll get breakdown of some of those receptors and fewer receptors. So that's upregulation. Downregulation happens in some cases, some types of hormones stimulate downregulation. In other words, you get a decrease over time. If you get long exposure to high levels of certain hormones, it can actually cause your cells to get desensitized to the hormone and downregulate or produce fewer receptors. And that seems bizarre, but it does happen. It's usually due to overexposure, like overproduction of a hormone. That overproduction of hormone actually causes sometimes downregulation, in which the target cells stop producing receptors, or they reduce the number of receptors, and it makes them less sensitive to the hormone. Now, certain glands, like the pancreas producing insulin and the pituitary producing growth hormone, instead of releasing constant, or long, releases of hormone, they will release it in pulses. They'll just release a burst of it. It's called pulsatile secretion, and instead of constantly releasing it, when it's needed. Now, that's supposed to help prevent downregulation, and it works most of the time. But unfortunately, with insulin, if you're every day eating lots of food, lots of calories, and especially lots of sugar, like you're drinking sodas, you're eating candy, and you're gaining weight, and then you're not exercising, so you're not using up that glucose with exercise, so your blood sugar is spiking, going up several times a day, and then your insulin levels spike throughout the day. Even though you do have this mechanism of pulsatile secretion, because you're producing so much insulin throughout the day in response to these spikes in your blood sugar, you can get downregulation of insulin receptors in your target cells. And that's what they're talking about when they talk about people being insulin resistant. What that means is all this insulin they've been producing over months of eating incorrectly and not exercising, overeating, and especially eating too much sugar and not exercising, that causes, even though you're getting bursts of insulin, you're getting too many bursts of insulin, and that causes downregulation of insulin receptors, and then your target cells, which are your body cells, don't respond to insulin like they should, so your blood sugar remains high, and you end up with type 2 diabetes. So usually type 2 diabetes, before a person has full-out type 2 diabetes, they have insulin resistance, which means there's some downregulation happening, and when they get actual type 2 diabetes, they've downregulated their receptors. Now, it can, if treated right away, if you start changing your diet, start eating better, eating less sugar, exercising and losing weight, if you do it soon enough, you can regain those receptors by, preventing those surges of sugar and surges of insulin. You can regain those receptors. But if you keep doing that and you don't stop the bad stuff that's going on, you don't change your diet, you don't exercise, over time, that type 2 diabetes cannot be reversed. But it is possible to reverse it in some cases, particularly if it's just, in the early stages of it. But if somebody lets it go too long, it can go from type 2 to type 1, where you become insulin dependent, because your pancreas just shuts down, stops producing insulin, just says, I give up, I'm not doing it anymore, and then you have to go on insulin, taking insulin to maintain your blood sugar. So downregulation, even with this, doesn't always work. Okay, so, or I should say, even with this, it does work, even, under extreme conditions like our bad American diet. That's an example. Okay, so, any questions about up and down regulation before we go on? So there are two kinds of hormones, lipid-soluble ones and water-soluble ones. Water-soluble ones can dissolve in water, in the blood plasma. Lipid-soluble ones can't, because they're lipid, they're non-polar, they don't dissolve in water, which is polar. Anything that's polar is going to be hydrophilic and will dissolve in water of the plasma. Anything that's lipid-soluble is non-polar, it's not going to dissolve very well in the plasma, and has to be attached to transport proteins in order to get from the gland to the target cell. This is similar to what happens with cholesterol and lipids. You have cholesterol in your blood is transported by transport proteins. That's why they call them lipoproteins. So when you're measuring your blood glucose, you're really measuring the level of lipoproteins, high density and low density lipoproteins. So anything that's lipid soluble and not water soluble, it has to be transported by proteins that can dissolve into the blood plasma and carry the lipid in the blood. So here's some examples of water-soluble hormones. Water-soluble ones are primarily amines, which are derived from amino acids, or short chain peptides, or polypeptides, which are short amino acid chains, not quite proteins. And then there's some protein hormones as well. Hormones like insulin is a hormone that's insulin and glucagon are protein hormones. So they don't have to be carried by a protein transporter. So you can see some examples here. Lots of hormones are water soluble and just released into the bloodstream. And you don't have to memorize all those different types of water soluble hormones, but anything that's water soluble doesn't need a protein transporter. And let me see, oops, that's not the one that's working. Okay, so in the case of water-soluble hormones, here's the hormone up here in the blood capillary, and the hormone is released out of the capillary into the interstitial fluid and on the target cell. This is the target cell membrane, there's the receptor. The water-soluble hormone binds to its specific receptor. So if this is insulin, this would be an insulin receptor. If it's glucagon, it's a glucagon receptor. Whatever the hormone is, you can look at any of those hormones on that list. Each one of those hormones that are in that list, each one of these has a specific receptor for it on its target cells. Each receptor is different. It only binds to a specific hormone. Once the hormone binds to the receptor, now if there's no hormone, there's no action. But once the hormone binds to its receptor, that hormone binding to the receptor activates a G protein. There's those G proteins again. Remember that some sensory neurons, like some of your taste receptor cells, they are what they call G protein coupled. In other words, in this case, binding of the hormone to the receptor activates a G protein, which in turn activates a second messenger system. And I'm not going to get into all the steps here, but a second messenger system like ATP being converted to cyclic AMP, which in turn sets into motion all this stuff. Notice that what ends up happening is a protein gets phosphorylated. That's usually what ends up happening. and you think, okay, well, big deal, protein gets a phosphate group added to it. Well, that in turn then activates other things to happen, and you get all sorts of metabolic responses to that phosphorylated protein. It sets into motion other physiological responses. So you don't have to memorize all these steps, but know that the receptor binding to the hormone in water-soluble hormones activates G protein, which in turn activates other metabolic processes. And those processes can be wide-ranging. Those reactions can be wide-ranging. So if this were insulin, and this were a regular, body cell, this G protein coupled messenger system would activate responses that would include putting more glucose transporters on the cell surface, which then in turn bring in more sugar. If this were a liver cell, it would stimulate that liver cell to respond by putting glucose together and storing it as glycogen. So, there's all sorts of responses that you can get depending on the target cell you're looking at and the hormone. Now, lipid-soluble hormones are nonpolar, and for example, steroid hormones. Now, this you should be, at least in terms of steroid hormones, you should know that those are lipid-soluble because these are all derived from cholesterol, and cholesterol is a lipid. It's a sterol lipid. And any hormone made from cholesterol that has this makeup, if you look at cholesterol, and I think I have a picture, here's cholesterol. Cholesterol consists of hydrocarbon rings, 4 rings that look like that, and then you got other parts stuck to it. That's cholesterol. Cholesterol is used to make all these other steroid lipids, all these other steroid hormones. Estrogen is made from cholesterol, testosterone, progesterone, corticosteroids like cortisol. Any kind of steroid hormone is a sterol lipid, and it's made from cholesterol. And if it's a lipid, it's mostly hydrocarbon, meaning it's nonpolar, hydrophobic, does not dissolve in water. So I think you should be able to figure out that that would be a lipid soluble or not soluble in water. Okay, if it's lipid soluble, it's not soluble in water. So this would be under that lipid soluble category. Now, I wouldn't necessarily, well, actually, when we get to thyroid hormones, you're going to learn about thyroid hormones, and you would need to know too that this is also lipid soluble, because we'll talk about that. You can't necessarily, looking at it, you might not be able to figure it out, but thyroid hormone, which there's actually 2 varieties of it, T3 and T4, those are lipid soluble and have to have transporters. So here's some examples. Vitamin D, you may not realize vitamin D is not only a sterol lipid made from cholesterol, but it is also acts as a hormone. It's a vitamin, it's also a hormone, it's a chemical messenger. So it falls in that category as well. So the way lipid-soluble hormones work, let me look at the time here. The way they work is here's the hormone, it gets picked up by a transport protein. The transport protein carries the hormone to the capillary. It releases the hormone goes to the cell, and instead of binding to a receptor on the cell surface, it diffuses through the lipid bilayer, through the phospholipid bilayer, into the cell. It moves into the nucleus and binds to a receptor in the nucleus. And this, in turn, this binding of the hormone to the receptor in the nucleus turns on a gene. The gene now makes messenger RNA, which in turn leaves the nucleus, goes to the ribosome, makes a protein, and that new protein might be an enzyme or it might be some other protein that then causes a change in the physiology of the cell, activates some response in the cell. So there's another picture that shows this as well. Here's the carrier protein, here's the target cell, here's the blood. The hormone goes into the cell. Sometimes the receptor protein is in the cytoplasm, sometimes the receptor protein is in the nucleus. It doesn't matter. The hormone binding to the receptor activates a gene, turns on a gene, which then is expressed as protein, and that protein causes a steroid hormone response. So all of sterol lipids work this way. So we'll pick up there on Thursday. I'm hoping I can get through all the hormones or most of them.